Hello, my name is Jessica Curran, and I want to share with you how I developed an alternative to chemical fertilizer made from local invasive seaweed. Being from Long Island, we know better than anyone else that we are known for our beaches. But the sand we were stepping on a decade ago is no longer the same, since now it's covered in layers of seaweed. And with our economy being so tourist dependent, this loss of beauty threatens the living conditions of the locals. In recent years, invasive species of rhodophyta, or red seaweeds, have taken hold of our coastal ecosystems, namely Daisophinia japonica and Gracilaria. These rhodophyta pose a serious threat since they use carbon dioxide as their main source of carbon intake, and as global warming and ocean acidification become more prevalent, increased PCO2 levels will allow them to outcompete native species and disrupt biodiversity. But red seaweed is made up of carbohydrates, proteins, and a wide range of nutrients that are needed for plant growth, indicating its potential as a fertilizer. And the main incentive for using seaweed fertilizer is to alleviate the harmful effects of chemical fertilizer, such as greenhouse gas emissions and eutrophication. In my research, I wanted to identify a solution to mitigate the effects of these invasive rhodophyta by repurposing them as a substitute for chemical fertilizer. I tested a variety of methods such as a soil drench, foliar spray, and agar medium on okra and Arabidopsis and identified its efficacy through growth, pigment concentrations, and distribution of nutrients. For my hypotheses, I predicted that the SLFs would perform better than a control with no added fertilizer, but less effective than a chemical fertilizer since it will likely have its nutrients in lower concentrations. I also predicted that the more concentrated SLFs would be most effective. To begin my experiment, I collected samples at local bodies of water on Long Island, specifically Shinnecock Bay and Peconic Bay. But the invasiveness of these rhodophyta has worsened so much that while I was collecting my D. japonica samples, I identified them in a location that previously were not there in a study in 2021, highlighting how much it's actually expanded its reach. For my fertilizer production, I dehydrated, boiled, strained, and centrifuged the samples to produce a liquid that was considered 100% SLF. And I did this separately for both my D. japonica and Gracilaria samples. To begin, I tested Gracilaria as a soil drench on okra in varying concentrations, and I compared these groups to a control with no applied fertilizer and a commercial NPK fertilizer. And for the other trials I ran, I likewise compared them to a control and commercial fertilizer. Regarding the final heights of okra, there was no significant difference, but the SLF had higher final heights, followed by control, and then NPK fertilizer, which I found surprising. Then I extracted the pigments using a 1-to-1 ratio of acetone to ethanol and identified it using a spectrophotometer. I was able to identify that the SLFs had higher concentrations than both the commercial NPK and control groups. I identified chlorophyll A, chlorophyll B, and carotenoids at its respective peaks, and I looked at these because it's a good indicator of plant health. And regarding the SLFs, they showed a dosage response having higher peaks of absorbance and thus higher concentrations of each pigment. Next, I used D. japonica as a foliar spray, which I chose since plants such as okra most efficiently take up nutrients through their leaves, and I was careful to choose small concentrations since leaves are easy to burn. SLF was shown to perform significantly better than the control and similar to chemical and growth factors such as final height, leaf length, and growth rate. I then identified chlorophyll A and B concentrations using a spectrometer and identified that the chemical fertilizer had significantly higher pigment concentrations than any other group, but the SLFs had higher pigments than the control. I then tested Gracilaria on Arabidopsis, and using a plant-based agar powder, I combined the agar solution with SLF, and compared these to a control with no fertilizer and another with commercial NPK fertilizer. One of the most important factors regarding plant health is identifying if they have adequate nutrients. Using the Tender Energy Spectroscopy beamline at Brookhaven National Lab, the Arabidopsis plants were scanned at 2400 EV and 4200 EV, which allowed me to identify phosphorus, sulfur, chloride, potassium, and calcium. Using PyXRF, I used a software that was allowed me to identify elemental peaks found in my samples. And the software also allowed me to produce maps to locate the elemental distribution of these nutrients. For potassium, I expected to see it located on the leaves since it's responsible for stomata opening, and the control sample shows a low distribution in the leaves, whereas the SLF groups show even distribution through the 1.25%, showing a more concentrated area through the axil, where a third leaf would be present. And this is especially important since the third leaf is formed by nutrients directly from this fertilizer. And the NPK fertilizers also had a high concentration at its leaf apexes, showing that both of these groups can effectively sustain plant growth. I then identified calcium, chloride, and sulfur, and at the and calcium was highly concentrated at the leaf apex for 5%, and it also showed the highest distribution of sulfur in the leaf, which is essential for chlorophyll formation and photosynthesis. And the 1.25 SLF indicated that it can support plant growth, shown by its third leaf development at the axil. I then tested phosphorus, which I expected to be located all throughout the plant since it's responsible for ATP creation and is a necessary limiting nutrient. All samples showed an even distribution throughout the plant, but the commercial NPK fertilizer showed a distribution towards the apexes of the leaves, and in each element test, overall in each element tested, the SLF was effective at providing these needed nutrients, which was comparable to the NPK fertilizer. Returning back to my hypotheses, we can determine that the rotophyta can be considered an effective SLF since it performed efficiently as chemical fertilizer regarding growth factors, pigment concentrations, and nutrient distribution. My solution of rotophyta derived seaweed offers three main incentives. It's effective since, as my research proved, it performed as efficiently as the chemical groups and better than that of a control. It's sustainable because it takes an otherwise invasive, harmful seaweed and it repurposes it as a substitute, which protects against the detrimental effects caused by chemical fertilizers such as greenhouse gas emissions and eutrophication. And arguably most important is that it's economical, making it a realistic solution. Its simple methodology allows it to be mass produced as a fertilizer, and there are tons and tons of free seaweed to be considered a free resource. This prospective solution should be considered as a serious alternative, as needed by our call to action to work towards environmental sustainability. Oh, I'm done? Okay.